Welcome to Module 3 of Anatomy and Physiology Systems 1, the Nervous System. Topic 3.1 will be an overview of the nervous system. If you open your workbook to Module 3, try to answer as many questions as you can as the mini-lecture progresses. The learning outcomes for Topic 3.1 are to obtain an overview of the nervous system and the tissues within it, to review action potentials and synaptic transmission, to list and name the classes of neurotransmitters and their functions. The nervous system is the master controller and the communicating system for the body. The cells communicate via electrical and chemical signals, which are rapid and specific, and cause almost immediate responses. So far, we have looked at the musculoskeletal system in Modules 1 and 2, and while the muscles contract to move joints, this movement occurs under the control of the nervous system. There are three main functions of the nervous system. The nervous system gathers information, which we call sensory input, via the millions of sensory receptors that monitor change both inside and outside the body. The nervous system is also responsible for processing and interpreting this information. Deciding what should be done in response to the sensory input is called integration. And finally, the nervous system's response to this information is termed the motor output. This includes activation of effector organs such as muscles and the glands. For example, you can see in this image that the sensory receptors of the eye gather information about where the glass of water is. This input is transferred to the brain via sensory neurons for processing and interpretation of the information. Once a response is decided upon, this information is then transferred by motor neurons out to a muscle in order to produce a response, such as contracting the biceps brachii to bend the elbow, bringing the glass of water towards the mouth for a drink. Sensory input is also known as afferent input. That is, information that is arriving at the brain for interpretation. Conversely, motor output, otherwise known as efferent output, information that is exiting the brain in order to elicit a response in the muscle. It might help to remember that afferent input is arriving at the brain, and that efferent output is exiting the brain. A, and E. Let's take a look at how the nervous system is organized. This diagram provides a great summary of the components of the nervous system and how they interact with each other. Throughout Module 3, we will progressively work our way through each of these subdivisions of the nervous system. The central nervous system, or CNS, consists of the brain and the spinal cord, which occupy the dorsal body cavity. The CNS is the integrating and control center of the nervous system. It is responsible for interpreting sensory input and dictating motor output based on reflexes, current condition, and past experiences. The CNS consists of bundles of axons traveling throughout the brain and spinal cord called tracts, and collections of neuron cell bodies called nuclei. The peripheral nervous system, or PNS, is that part of the nervous system outside the CNS, so everything else. In the PNS, bundles of axons exiting from the brain and spinal cord are called nerves. The collections of neuron cell bodies are called ganglia. These peripheral nerves provide the communication lines that link all parts of the body to the CNS. Functionally, the peripheral nervous system has two subdivisions. The sensory or afferent division conveying information to the central nervous system from the sensory receptors located out in the periphery. And the motor or efferent division, which transmits impulses away from the CNS to the effector organs, the muscles and the glands. This motor division of the PNS is further subdivided into the somatic nervous system, otherwise known as the voluntary nervous system, which activates skeletal muscles, and the autonomic nervous system, otherwise known as the involuntary nervous system, which regulates the activity of smooth muscles, cardiac muscles, and glands. And finally, the autonomic nervous system also has two subdivisions. The sympathetic nervous system, which mobilizes all of the body systems during activity, is known as the fight and flight response system. The parasympathetic division, which is responsible for conserving energy, is commonly known as the rest and digest system. Before we take a look at these systems over the following few mini lectures, let's start by reviewing the tissues involved in the nervous system. There are two kinds of nervous tissue: neuroglia and neurons. Neuroglia wrap around the more delicate neurons, providing them with support and protection. The neurons are excitable nervous tissue which respond to stimuli and transmit electrical signals from one part of the nervous system to another. For each neuron found within the nervous system, there are approximately 10 neuroglia. Within the CNS, the neuroglia include astrocytes, microglia, ependymal cells, and oligodendrocytes. In the PNS, they include satellite cells and Schwann cells. Neurons may be classified according to how many processes extend from the cell body. They are described as unipolar, bipolar, or multipolar neurons. The first two types of neuroglia found in the CNS are astrocytes and microglia. Astrocytes are the most abundant of the nervous tissue found in the CNS and are shaped like stars. Their radiating processes cling to the neurons in order to anchor them to nearby capillaries. Their feet to help determine capillary permeability, and they guide formation of synapses or junctions between neurons. Astrocytes also mop up leaked potassium ions and recapture neurotransmitters released within the nervous system. Microglia are the defense cells in the CNS. They have long, thorny processes that monitor the health of nearby neurons, so that if they detect damaged or necrotic tissue, they migrate towards the injured neurons. They then transform into a specialized macrophage that is able to phagocytose microorganisms. Ependymal cells are also a type of neuroglia found within the CNS. These cells line the central cavities of the brain and the spinal cord and form a permeable barrier between the cerebrospinal fluid and tissue fluid around cells. Fine cilia or hair-like projections on the ependymal cells help circulate the cerebrospinal fluid. Oligodendrocytes form protective and insulating myelin sheaths around the thicker nerve fibers in the CNS. They are able to myelinate several nerve fibers at once. These cells are destroyed in patients with multiple sclerosis. The two types of neuroglia found within the peripheral nervous system are called satellite cells and Schwann cells. Satellite cells surround cell bodies of neurons as support cells. They have similar functions to the astrocytes found in the CNS. Schwann cells surround all the thicker nerve fibers in the PNS and are responsible for producing the myelin sheath, much like the oligodendrocytes found in the CNS. The Schwann cell plays a vital role in regeneration of damaged peripheral nerves. In addition to neuroglia, the nervous system is made up of billions of neurons, which are the nerve cells, the structural units of the nervous system. They conduct messages from one part of the body to another via nerve impulses. Neurons are amitotic, which means once they assume their role as a communication link within the nervous system, they lose their ability to divide. Unfortunately, this means that neurons can't be replaced if they are destroyed. However, there are exceptions. Some stem cells contained in the lining of our nose in the olfactory epithelium and in the hippocampus of the brain have been found to produce new neurons throughout life. Luckily, neurons have extreme longevity. Most of them can function optimally for a lifetime, provided they are given good nutrition. They also have a high metabolic rate, which means they can't survive more than a few minutes without oxygen and glucose. The typical structure of a motor neuron is seen in this image. It consists of a cell body which contains a nucleus, arm-like processes extending from the cell body called dendrites, and a long, slender process called an axon. This axon is insulated by a myelin sheath produced by the Schwann cells, which help to increase the speed of conduction of the action potential from its production at the axon hillock to its end at the axon terminals. Different neurons throughout the body can be classed structurally as either multipolar, bipolar, or unipolar, depending on the number of processes extending from the cell body. A multipolar neuron has multiple processes extending from the cell body, all of which are dendrites, except for a single axon. Examples of multipolar neurons are the motor neurons that conduct impulses along different pathways from the CNS to the skeletal muscles. Bipolar neurons have a cell body with two processes extending from it. One is a fused dendrite and the other is an axon. Bipolar neurons are specialized sensory neurons that are located within the sense organs, such as those in the retina of the eye, that are responsible for transmitting visual input from the eye to the brain. The unipolar neuron has a cell body with a single process. However, this single process divides into a peripheral process and a central process that together comprise one axon. An example of a unipolar neuron is the sensory neuron that conducts afferent impulses towards the CNS for interpretation. So we have sensory neurons traveling toward the brain, unipolar neurons. We have motor neurons traveling away from the brain, multipolar neurons, and in between we have interneurons, or association neurons, that lie between the motor and sensory neurons in the neural pathway. But how do we get the message from one neuron to another? How do they talk to each other? This slide represents the assumed knowledge you have for this course. If resting membrane potentials, graded potentials, and action potentials are unfamiliar to you, please review these concepts in your textbook. It is important to remember that nerves have excitable membranes at rest. The voltage or charge difference across the cell membrane is -70 millivolts. At rest, the outside of the cell is more positively charged, and inside the cell is more negatively charged. If possible, charges pass into the cell, and the resting membrane potential becomes more positive, or less negative, towards zero, and reaches the threshold for firing an action potential. The cell membrane depolarizes, and a nerve impulse is conducted to the very end of the axon, where it can then communicate with the next neuron, or the neuromuscular junction, in a case of a muscle cell. The junction where one neuron connects with the next is called a synapse. Synapses come from the Greek word syn, meaning to join. It is the junction that allows information transfer from one neuron to the next, or from one neuron to an effector cell. That's where all the action happens. The neuron conducting the impulse towards the synapse is called the presynaptic neuron. The neuron transmitting the impulse away from the synapse is called the postsynaptic neuron. That is, at any synapse, the presynaptic neuron sends the information, and the postsynaptic neuron receives the information. Neurons are aligned end to end, or side to side. Remember that pre means before, post means after, and synapse means junction or connection. There are two different types of synapses, electrical and chemical. Chemical synapses are the most common type and are specialized to release and receive chemical messengers known as neurotransmitters. In the presynaptic neuron, the axon terminal contains many little sacs called synaptic vesicles, each containing thousands of neurotransmitter molecules. The neurotransmitter receptor is located on the postsynaptic membrane, which may be on a dendrite or another cell body, sometimes referred to as the soma. Let's review how the message is transferred from the presynaptic neuron across a chemical synapse to the postsynaptic neuron. Nerves are electrically excitable, and if a change in the membrane potential reaches threshold, an electrical charge travels along the axon of the neuron in the form of an action potential. Transmission of nerve impulses along an axon and across the chemical synapses depends on the release, diffusion, and receptor binding of the neurotransmitter molecules. Presynaptic and postsynaptic neurons are separated by a synaptic cleft, which allows the current to be dissipated. This prevents a nerve impulse from being directly transmitted from one neuron to another. You might remember in Module 2 we discussed the neuromuscular junction, which is a specialized chemical synapse. Briefly, the chain of events that occurs at the neuromuscular junction is as follows. Firstly, the action potential arrives at the axon terminal. Secondly, the voltage-gated calcium channels open, and calcium enters the axon terminal during the brief time the calcium channels are open. Calcium floods down the electrochemical gradient from the extracellular fluid into the terminal. Thirdly, the surge of the calcium into the axon terminal causes fluid-filled bubbles called synaptic vesicles to release neurotransmitters by exocytosis. That is, the synaptic vesicles fuse with the presynaptic membrane and empty their contents into the synaptic cleft. Calcium is then quickly removed from the terminal, either via the mitochondria or by an active calcium pump. Next, the neurotransmitters diffuse across the synaptic cleft and bind to a specific receptor on the postsynaptic membrane. When the neurotransmitter binds to the receptor protein on the postsynaptic membrane, the receptor changes shape. This opens the ion channels and creates graded potentials. Depending on the receptor protein to which the neurotransmitter binds and the type of channel the receptor controls, the postsynaptic neuron may be either excited or inhibited. For as long as the neurotransmitter is bound to the postsynaptic receptor, it will continue to affect membrane permeability and block the reception of additional signals from presynaptic neurons. Therefore, it's important that the neurotransmitter is removed to wipe the slate clean. This may happen in one of three ways. Either the neurotransmitter diffuses away from the synapse, or it is degraded by enzymes present in the synaptic cleft, or there is reuptake of the neurotransmitter by astrocytes or the presynaptic terminal where the neurotransmitter is stored. The neurotransmitter receptors mediate local changes in the postsynaptic membrane potential that are graded. They vary in strength based on the amount of neurotransmitter released and how long it remains in the area. As mentioned on the previous slide, chemical synapses are either excitatory or inhibitory, depending on how they affect the membrane potential of the postsynaptic neuron. An EPSP, or excitatory postsynaptic potential, occurs at an excitatory synapse where binding of the neurotransmitter depolarizes the postsynaptic membrane. This means that if enough neurotransmitter binds, the membrane potential becomes more positive and local graded depolarization occurs. This is very short-lasting, and then the membrane returns to its resting potential. These currents run along dendrites or the cell body and decline with distance. However, they can often spread all the way to the axon hillock. The function of the excitatory postsynaptic potential is to help trigger an action potential, at this point if the membrane potential is strong enough. That is, a postsynaptic membrane potential that is more positive is more likely to fire. Inhibitory postsynaptic potentials occur with hyperpolarization of the postsynaptic membrane. That is, when neurotransmitters bind at inhibitory synapses, they reduce the postsynaptic neuron's ability to generate an action potential. The inside of the membrane becomes more negative, decreasing the membrane potential, making it less likely that the neuron will fire. Larger depolarizing currents would be required to induce an action potential. Where there is a lot of information on this slide, the most important concept to understand is contained in the box at the bottom of the slide. The factors that affect synaptic transmission are the amount of neurotransmitter released, the destruction or blocking of receptor binding, or the reuptake of neurotransmitters. In the next few slides, we will examine a few of the neurotransmitters and see how function is altered if receptor binding is blocked or destroyed or reuptake is altered. Along with the electrical signals, neurotransmitters are the means by which the nervous system communicates messages to the rest of the body. Although some neurons produce and release only one kind of neurotransmitter, most make two or more, and the coexistence of more than one neurotransmitter in a single neuron makes it possible for the cell to exert several different influences. Neurotransmitters may be classified chemically or functionally. We will take a look at some of the following neurotransmitters that are classified by chemical structure. Acetylcholine, the biogenic amines such as noradrenaline, or as it is known in other parts of the world, norepinephrine, dopamine, serotonin, and histamine. Amino acids such as gamma-aminobutyric acid, or GABA, and glutamate, and peptides such as endorphins. These next few slides are filled with information from the tables in your textbook. However, I want the focus on neurotransmitters to be less about their chemical structure and more about their function. Acetylcholine, or ACH, was the first neurotransmitter discovered. ACH was thought to be important in skeletal muscle contraction when it was found concentrated at the neuromuscular junction in frogs' legs. After injecting ACH into the junction, the frog muscles spontaneously contracted. ACH is synthesized from acetic acid and choline, and is transported into synaptic vesicles for later release at the neuromuscular junction. Once released at the presynaptic terminal, ACH binds briefly to the postsynaptic receptor and is then released and degraded by the enzyme acetylcholine esterase at the synaptic cleft. ACH is released by all neurons that stimulate skeletal muscles and many neurons of the autonomic nervous system. It always has an excitatory effect at the neuromuscular junction of skeletal muscle, where it binds to nicotinic receptors. However, its effect may be excitatory or inhibitory, depending on the type of muscarinic receptor on the visceral effector organs. So, the end result is, depending on which receptor ACH binds to, it will have a different effect. But what happens if the release of ACH from the presynaptic neuron into the chemical synapse at the neuromuscular junction is prevented? This is precisely how botulism toxin, or Botox, works. Injected botulinum toxin blocks the release of ACH from the presynaptic neuron, which prevents the muscle contraction from occurring, resulting in muscle paralysis. Botulinum toxins now play a significant role in the management of a wide variety of medical conditions, such as focal dystonias, which involve involuntary muscle contractions causing sustained abnormal postures, hemifacial spasms, spasticity associated with cerebral palsy, and headaches. Botox is also commonly used for cosmetic corrections of lines, creases, and wrinkles on the face, chin, and neck. If the release of ACH is prevented, it is no longer excitatory. It can't cause skeletal muscle to contract and produce a frown, and the wrinkles on the forehead are avoided. Neurotransmitters contained in the biogenic amines group have an amine, the NH2 group, attached to the end. These include noradrenaline, or norepinephrine, dopamine, serotonin, and histamine. In the next slide, we will take a closer look at noradrenaline and dopamine, two of our feel-good neurotransmitters, known as catecholamines. You'll notice how similar their chemical structure is in the green shaded boxes on the screen. The catecholamines are broadly distributed in the brain, are heavily involved in emotional behavior, and help regulate the biological clock. They are both generally excitatory, feel-good neurotransmitters, and imbalances of these neurotransmitters are associated with mental illness. For both noradrenaline and dopamine, reuptake of the neurotransmitters back into the central nervous system is blocked by cocaine and antidepressants, or SNRIs, serotonin noradrenaline reuptake inhibitors. This means that the neurotransmitters remain in the synaptic cleft longer, where they are able to continue exerting their influence, so you continue to feel good for longer. The excitatory effect of noradrenaline is enhanced under the influence of amphetamines or pseudoephedrins, such as used to be found in the Codral Cold tablet. The excitatory effect of dopamine is enhanced in the presence of L-DOPA or levodopa, which is the drug prescribed to patients with Parkinson's disease. Dopamine is deficient in Parkinson's disease, whereas increased dopamine neurotransmission occurs in schizophrenia. Another classification of neurotransmitters are the amino acids. Amino acids contain both an amine group, NH2, and a carboxylic acid group, COOH. The main inhibitory neurotransmitter is the classification of GABA, gamma-aminobutyric acid. GABA is found in the cerebral cortex, the hypothalamus, the cerebellum, and the spinal cord. This is the main inhibitory neurotransmitter in the brain, and its inhibitory effects are enhanced by alcohol and anti-anxiety drugs such as Valium. Alcohol stimulates GABA release, which inhibits the postsynaptic neurons from firing. That is, an IPSP is elicited. The result is poor motor coordination, a decreased heart rate, and sleepiness. The last group of neurotransmitters that we are interested in are the neuropeptides, which are essentially strings of amino acids held together. These are the endorphins, which include dynorphin and enkephalin. These neurotransmitters are generally inhibitory and are distributed widely throughout the brain, including the hypothalamus, the pituitary gland, and the limbic system, as well as throughout the spinal cord. The endorphins are our natural opiates. They inhibit pain by inhibiting the effect of substance P, which is an important mediator of pain signals. Endorphin release is enhanced when an athlete gets a second wind, and can probably be deemed responsible for the runner's high. Release of endorphins in the spinal cord blocks the message of discomfort coming from the periphery, like "my feet hurt" while running in a marathon. Endorphins are incredibly addictive. They make you feel good. The effects of the endorphins are mimicked by morphine, heroin, and methadone, as they bind to the same receptors as endorphins. However, the opiate high from morphine and heroin is even more intense, but wears off quicker after each successive use, leaving the user craving the next high sooner each time. We have now achieved all three learning outcomes for Topic 3.1, an overview of the nervous system. This brings us to the end of the first mini-lecture in Module 3.